Good morning, students. You are all welcome in this new session of 12th Science lectures. We already started these lectures previously, online lectures, to overcome that problem. Means we cannot go at the college, but still we are teaching and learning at home by this new method. So, we already started that first chapter. That is reproduction in lower and higher plants. Okay, and just a small topic is remaining. So today we'll complete that chapter, and the last topic was seed and fruit development. The goal of reproduction in every living organisms, including plants, is to create offsprings for the next generation. It is very obvious that the goal of reproduction is to produce offsprings, including in plants also, in animals also. Okay, one of the ways that plants can produce offsprings is by forming or making seeds. Okay, so in plants there are so many different methods to produce new plants, just like vegetative propagation, budding, okay, zoospore formation, and all that. We already discussed all that things. So one of the method is to produce seeds. The flowers must be pollinated in order to produce seeds and fruit. Hmm. So for production of seeds, the flower must be pollinated in order to produce seeds and fruits. This means that flower pollination is very essential, and without that we cannot see the production of seeds and fruits. So seed development is initiated by fertilization. You should revise that topic, post fertilization changes. So in that topic we discuss how the seed formation takes place. After fertilization there is a male union of male gamete with the female gamete, and then after some period there is the formation of seeds. So the integuments of the fertilized ovule persist, means whatever the integuments are there in that anatropous ovule that are remain up to seed formation, these integuments are converted into seed coats. Outer integument and inner integument, both these integuments are converted into seed coat. Hmm? The integuments of the fertilized ovule persist and get transformed into the seed coat of mature seed. So up to this we already discussed. Now what is next? See this diagram. This white colored portion, which is occupied most of the area in this dicot seed, is called as cotyledon. It is the structure of bean seed. ओके, घेवडाची जी काही बी असते त्याचं हे स्ट्रक्चर आहे. It is the LS, longitudinal section of bean seed, and this white colored structure is called as cotyledon. Now the outer covering of this seed is called as seed coat, okay, which is initiated from the integuments of seeds. After that, this portion, which is white colored portion, it is, you should observe this structure carefully. This structure is called as radicle, by which root formation will take place. Now this green colored outgrowths, two outgrowths, this is called as epicotyl. Epicotyl, and below that there is the hypocotyl. You know that epi means upon, means it is the above structure, and below that there is the hypocotyl. Okay, it is the radicle, and this whole structure is called as embryo. So it is the structure of dicot seed. Now we'll discuss monocot seed. See, it is the structure of monocot seed, maize seed. Monocot, single cotyledon is there. Just like dicot, there is also outer covering is seed coat, and also it is called as fruit wall. After that, these two parts are there because of this line layer. This layer is called as aileron layer. It is present at the whole structure of this endosperm. Okay, so this endosperm is called as aileron layer, and below that it is the larger portion, and below that there is the smaller portion which is called as scutellum. Okay, means monocot seed is divided into two parts by this aileron layer. The bigger portion is called as endosperm, while the lower portion is called as scutellum. Okay. After that, this is the plumule and it is the radicle. From plumule, stem formation or shoot formation will take place, and from radical, root formation will take place. Okay, so this radical is covered by this coleorhiza, and this plumule is covered by coleoptile. Covering of the plumule is called as coleoptile, while the covering of the radical is called as coleorhiza. So, like this, there is the structure of maize seed. Now, next, seed sometimes consists two distinct coverings. A typical outer seed coat, the testa, and the inner thin membranous is tegmen. Okay, so you have seen that in anatropous ovule there are two layers, that is outer covering outer integument and inner integument. Outer integument is called as outer integument is converted into testa, while the inner integument will convert into tegmen. So testa is outer covering of the seed which is thick, and below that there is the inner covering which is membranous or papery or somewhat thin. Okay, एखाद्या पापुद्रासारखी, that is the tegmen. Okay, now next, the nucellus in the ovule may persist in some genera like black pepper and beet as a thin papery layer. The perisperm means the plant genera like black pepper, काळी मिरी जे आपण म्हणतो, आणि beet, in this type of plants nucellus in the ovule may persist. Nucellus may be there, means it remains there in the form of thin and papery layer, that is called as perisperm. The outer covering is very thin and papery, it is called as perisperm. In some seeds, the food reserves in the endosperm are partially used up in the development of an embryo. So, in some seeds, some other seeds are there in which reserve food material is present in the endosperm and it is partially used up for the formation of embryo. Okay. Obviously, in such seeds, the endosperm remain conspicuous and fills a greater part of the seeds. This means the cases or the seeds in which reserve food material is partially used up, in this type of seeds, there is the endosperm remains conspicuous, means it is very remarkable and it fills the greater parts of the seeds. For example, thus the resultant seeds is endospermic or albuminous seeds. Means, in short, the seeds in which endosperm remains after maturity also, that is called as endospermic or albuminous seeds. For example, seeds of castor, coconut, maize, etc. Okay. Next, in other seeds, embryo absorbs food reserved from the endosperm and completely during its developmental stages. Means, in other type of seeds, this endosperm is completely absorbed by the embryo. Okay, embryo absorbs reserved food from the endosperm completely during its developmental stages. Thus, endosperm disappears in the mature seeds. Okay, just now we have seen that in coconut or maize or castor, endosperm remains there after maturity also. That is called as endospermic or albuminous seeds. While in other type of seeds, endosperm, whatever the nutritive tissues are present in the endosperm are completely absorbed by the embryo. Okay, during its developmental stages. Thus, endosperm disappears or disorganizes in the mature seeds. The resultant seed is non-endospermic or ex-albuminous seeds. For example, pea, bean, etc. Okay, you can compare maize with the pea. In case of maize, there is a endosperm or nutritive tissues remain there. That is called as endospermic or albuminous type of seeds, while such seeds like pea or bean, endosperm completely disappears. Okay, and that's why it is called as non-endospermic or ex-albuminous seeds. The cotyledons in the some Non-endospermic seeds act as food storage, and in others, they are first photosynthetic organs. Okay, whatever the cotyledons, in some non-endospermic seeds, they act as food storage. Cotyledons are used as the storage of food material, and in some other seeds, they are first photosynthetic organs. कोटिलिडॉनचे दोन रोल इथे आपल्याला दिसतात ते फर्स्ट इट ऍक्ट्स ॲज फूड स्टोरेज टिश्यूज while in some others it acts as first photosynthetic organs. Okay, micropyle persist as a small pore in seed coat to allow the entry of water and oxygen during soaking. जर आपण पाहिलं की काही जे काही सीड्स आहे किंवा मायक्रोपाईल पर्सिस्ट मीन्स शेंगदाणे वगैरे जर आपण पाहिलं आणि ते जर पाण्यात भिजवले तर स्मॉल ओपनिंग इज देअर ॲट कॉर्नर वन कॉर्नर दॅट इज मायक्रोपाईल. इट पर्सिस्ट ॲज अ स्मॉल पोर ओके इन groundnut. So micropyle persists as a small pore in seed coat to allow the entry of water and oxygen during soaking. Okay, next. In gram seeds also हरभरे जर आपण पाण्यात भिजवली, so at that time micropyle persists there as a small pore and in the seed coat to allow the entry of water and oxygen during soaking. Now, fruit development is triggered by hormones produced by developing seeds. As mentioned earlier, after fertilization, the zygote is formed and the ovary begins to differentiate into the fruit and ovary wall develop into pericarp. This pericarp is basically three layered which get differentiated in the fleshy fruit like mango, coconut, etc. Okay, so fruit formation is triggered by hormones by developing seeds. Whatever the seeds are present inside that fruit that triggers some hormones for food development. Okay, after fertilization, the zygote is formed. Post fertilization changes मध्ये आपण पाहिलं की there is the zygote formation takes place after fertilization, and the ovary begins to differentiate into the fruit, and ovary wall develops into pericarp. That ovary becomes fruit, and the ovary wall becomes pericarp, that is outer covering of that fruit. Now, pericarp is basically three layered structure. In the mango, we have seen this pericarp is divisible into three layers, that is outer epicarp, middle mesocarp, and inner endocarp. Okay, so pericarp is basically three layered which get differentiated in fleshy fruit like mango and coconut. We have seen that diagram. Okay, next, what are the parts of the fruit? That is, just now we have seen pericarp and seed. In some fruits like mango, the pericarp is again divisible into three layers, that is outer epicarp, inner mesocarp, and innermost is the endocarp. Okay, so first part of the fruit is pericarp, and inner to that there is the seed or seeds. So this is the answer of this first question. Second, what is the difference between true fruit and false fruit? It is also we have discussed. True fruit means the fruit which develops from the ovary, that is called as true fruit, and the fruits which develop from the other parts of the flower other than ovary, ओव्हरीच्या व्यतिरिक्त इतर पार्ट्स पासून जेव्हा फ्रूट फॉर्मेशन होत असेल, that is the false fruit. Okay, significance of seed and fruit formation. First, fruits provide nourishment to the developing seeds. Okay, whatever the seeds, the main organ of reproduction in plants, that is the seed. So these fruits provide nourishment to these developing seeds which are present inside that fruit. Second, fruits protect the seeds in immature condition. Okay, if some means at the early stages of development, when that seeds are in immature condition, at that time fruits protects that seeds. Next, seeds serve as important propagating organs, units of plant. They serve as important propagating organ, means near about 70 to 80% of the plants produce because of seeds. So seeds, the third significance of the seed is it serves as the important propagating organ or unit. Next, Seeds and fruits develop special devices for their dispersal and thus help in distribution of the species. Okay, seeds and fruits, various modes of dispersal of these seeds by wind or by animals. Okay, so seeds and fruits develop special devices. Special devices are there for their dispersal and distribution, and so that the species will develop and distribute. Next, the dormancy. What is dormancy? It is the state of metabolic arrest that facilitates. Dormancy is the state of metabolic arrest that facilitates the survival of the organism during adverse environmental condition. Dormancy means what? Even though there are some reverse or adverse environmental condition at that time also, that organism is having power to survive in that adverse environmental condition. So it is the metabolic arrest. Dormancy is the metabolic arrest that facilitates the survival of that organisms during adverse environmental condition. Structural or physiological adaptive mechanism for the survival is called as dormancy. You should underline this definition, that is structural or physiological adaptive mechanism for the survival is called as dormancy. If that organism is having some adaptive mechanism or at that time, there are some structural and physiological adaptive mechanisms in that plants for their survival is called as dormancy. Mature and viable seeds will not germinate even in presence of the favorable condition, and they are dispersed at different places during dormancy. Some even though some mature and viable seeds are there, then they cannot germinate even there is a favorable condition. So some problems are there at that time. They are dispersed at different levels places by dormancy. Viable seeds germinate only after completion of dormancy period. So without completing the dormancy period, the seeds cannot be dispersed or germinate. Now apomixis. Apomixis means it is the phenomenon of formation of embryos through asexual method of reproduction without formation of gametes and the act of fertilization. Apomixis means fruits are formed without fertilization, that is called as apomixis. It is the phenomenon of formation of embryos through asexual method of reproduction without the formation of gametes, means uh, fertilization without formation of gametes is called as apomixis. Alternately, it is unusual sexual reproduction where there is no meiosis and syngamy. No meiosis takes place, no any syngamy. Okay. Embryo develops in the ovule and ovule develops to form seeds. Directly embryo develops into ovule. No any middle steps are there. So embryo develops in the ovule and ovule develops into seed is called as apomixis. Apomixis. In apomixis, when a gametophyte organ or cell produces embryo-like structure without fertilization, that it is called as apogamy. Okay, when that gametophyte organ or cell produces embryo-like structure without fertilization. Just now I told you that when there is a fertilization is there, but gamete formation is not there. Okay, so when gametophyte organ or cell produces embryo-like structure without fertilization, then it is called as apogamy. Similarly, when diploid sporophyte cell produces a diploid gametophyte without undergoing meiosis is called as apospory. So you should list these terms: apomixis, or before that we have seen this dormancy, apomixis, after that apogamy and apospory. Okay, In apomixis, when gametophyte organ or cell produces embryo-like structure without fertilization, some embryo-like structure is there, but it is formed without fertilization. It is called as apogamy. And when diploid sporophyte cell produces a diploid gametophyte without undergoing meiosis, it is called as apospory. Hmm? Without fertilization, apogamy, and without meiosis, it is called as apospory. For example, in orange or mango. The main categories of apomixis are: first, recurrent apomixis. In this type, the embryo sac generally rises either from archisporial cell or from some other part of the nucellus. Okay, embryo sac generally form either from archisporial cell or some other part of the nucellus in recurrent apomixis. Diplospory, the unreduced embryo sac, is derived from the diploid megaspore mother cell. For example, taraxamum, sorry, taraxacum. In apospory, nucellar cells gives rise to the apomictic embryo sac. So it is the recurrent apomixis. Apospory nucellus gives rise to the apomictic embryo sac. Non-recurrent apomixis means in this type, megaspore mother cell undergoes usual meiotic division as usual and haploid embryo sac is formed. Okay, means it is same like uh, the method which we have already studied. Here, the embryo arises either from the egg by parthenogenesis or from some other haploid cells of gametophyte through apogamy. Plants produced by this method are generally sterile. So these plants are sterile and they do not reproduce sexually. For example, Nicotiana. Hmm? Non-recurrent apomixis. So up to this it is same, but the embryo arises either from the egg or by parthenogenesis, means development without fruit formation. So this or from some other haploid cells are there of gametophyte through apogamy, and hence they are sterile. Now adventive embryony. In this type, embryos may develop from somatic nucellus or integuments along with the normal zygotic embryo. It is common in mango, orange, lemon, etc. This gives rise to a condition called polyembryony. Okay, so here embryos may develop from somatic nucellus. No, there is no any gamete formation. Directly the nucellus cells are utilized by that embryos or also from the integuments, okay, along with the normal zygotic embryo. So the examples are mango, orange, lemon, etc., and is used as the condition called as polyembryony. Genetically identical plants can be produced effectively and rapidly by apomixis. So it is the advantage of this method. That's why the name is adventive polyembryony. Parthenocarpy. In this term, this term is coined by Knoll in 1902. It is the condition in which fruit is developed without the process of fertilization. Hmm? When the fruit is formed without the process of fertilization, then it is called as uh, parthenocarpy. It occurs naturally in some varieties of pineapple, banana, papaya, etc. Parthenocarpy can be induced. Okay, when the fruits are formed without fertilization, then it is called as parthenocarpy, and it is occurs naturally in some plants like pineapple, banana, or papaya. In these plants, it seems that the placental tissue in the unfertilized ovary produce auxin. It is the name of hormone, growth hormone, IAA, indole 3 acetic acid. IAA means indole 3 acetic acid, which is responsible for enlargement of ovary into fruit. The fruit resembles the normally produced fruit, but it is seedless. Means it looks like normal fruit, but it is seedless fruit. Hmm? This means that the parthenocarpic fruits are seedless. Wait for a minute. So this point is already over. Now next, polyembryony. It is the development of more than one embryos inside the seed, and the condition is described as polyembryony. The name itself suggests the meaning, that is polyembryony. Inside the it is the development of more than one embryos inside the seed. Okay, it was first noticed by Leeuwenhoek in the seeds of citrus genus, means Limbu jahel, citrus genus. So Leeuwenhoek is the scientist who first observed these seeds inside the citrus. It is the occurrence of more than one embryo in a seed, which consequently results in the emergence of multiple seedlings. Okay, this means that by a single seed you can get more plants, more seedlings, multiple seedlings. So it is the occurrence of more than one embryos in a seed, which is consequently results in the emergence of multiple seedlings. Okay, now the additional embryos result in the form of differentiation and development of various maternal and zygotic tissues associated with the ovule seed. Additional embryos, where it comes from? It comes from the differentiation and development of various maternal and zygotic tissues which are present inside the ovule. Okay, polyembryony may be true or false depending upon whether many embryos arise in the same embryo sac or in the different embryo sacs of the same ovule. So this polyembryony may be true or false. It is dependent upon the embryos, many embryos arising in the same embryo sac, then it is true, or they are present in the different embryo sacs in the same ovule, then it is false polyembryony. In adventive polyembryony, an embryo developed directly from diploid cell of the nucellus and integuments, as in citrus. In cleavage polyembryony, so first is adventive polyembryony. An embryo developed directly from the diploid cell of nucellus, okay, and integuments, as in citrus. But in cleavage polyembryony, zygote proembryo sometimes divides into many parts or units. Each unit then develops into an embryo. Okay, so polyembryony increases the chances of survival of new plants. Nucellar adventive polyembryony is of great significance in horticulture. So in horticulture branch, it is very adventive. Okay, cleavage polyembryony. In this type, zygote proembryo sometimes divides into many parts or units. Okay, and each unit is developed into embryo, and each embryo will give rise to the new plants. Okay, now see, do you know parthenogenesis is the development of embryo directly from the egg without fertilization. Okay, directly from the egg cell or a male gamete. It is a kind of apogamy. It is the type of apogamy. Second, agamospermy. Agamospermy, here the plants produce seeds, but embryo inside it is produced without meiosis and syngamy. Agamospermy. Plants produce seeds, but embryo inside the plant is produced without meiosis or syngamy. It cannot, it not going into the cell division. It do not goes the type of germ cell division, that is meiosis. Parthenocarpy can be induced artificially by spraying gibberellins. It is the growth hormone, delaying pollination, use of foreign pollens, etc. So purposefully, manually parthenocarpy can be induced in horticulture department by spraying gibberellins. It is the type of growth hormones. Also by delaying pollination, जर pollination delay केलं तरी parthenocarpy induce होतंय तिथे, and use of foreign pollens. जर बाहेरून pollens तिथे आपण डिपॉझिट केले तर. So these are some artificial methods of parthenocarpy to induce parthenocarpy in the plants, flowering plants. Next, genetically uniform parental type seedlings are obtained from nucellar embryos. जे genetically uniform parental parents असतात. So genetically uniform parental type seedlings are obtained from nucellar embryos. So they are obtained from the nucellar embryos. So here we ends with this chapter, and your homework is try to solve all exercise question answers as well as the questions which are there inside the that chapter. Okay, thank you.